Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test CUCET or CUET MSc Entrance question paper for the subject Plant Biotechnology conducted in the year 2022. The code was PGQP78. So I have discussed here only the biology questions. So coming to the first question, assertion and reason. So first assertion, somatic hybridization is an excellent method to combine two genome bypassing sexual reproduction. That's correct. It can be used to produce symmetrical allotetraploids. That is also correct. So the answer is option 1. Both A and R are true, and R is the correct explanation of A. So that is correct. The next one. Organ specific secondary metabolite product is best achieved by. So it is best achieved by hairy root culture. So answer is option 2. The next one. Given below are two statements. One is labeled as assertion and the other is reason. Nitrogenase enzyme is sensitive to oxygen and must be protected from oxygen mediated inactivation. That is correct. An oxygenic condition is maintained by leg hemoglobin and facilitates symbiotic nitrogen fixation. So nitrogenase enzyme is involved in nitrogen fixation in the root nodules, and it is very sensitive to oxygen. So an oxygenic condition is provided by the leg hemoglobin. So both A and R are true, and R is the correct explanation of A. Answer is option 1. Which of the following statement is not true about artificial or synthetic seed? Somatic embryos are encapsulated with calcium alginate. That's correct. Calcium alginate is not damaging to somatic embryos. That is also correct. Cobalt alginate capsule is delicate and require careful handling during transportation and storage. That is not correct, and that is your answer. Calcium alginate can be incorporated with nutrients and growth regulators. That is also correct. So coming to the next question. Forced axillary bud proliferation is induced by the exogenous application of. So axillary bud proliferation, if you want, you have to apply cytokinin plant hormone. So the answer here is option 2. The next one. Given below are two statements, 1 and 2. So statement 1 is organogenic competent cells can be induced to form a specific organ. That is correct. Once induced to organogenesis, the cells become irreversibly committed to form specific organ even if the inductive hormone is withdrawn. So both statements 1 and 2 are correct. Answer is option 1. Then next one. Statement 1. Initiation of embryo development is stimulated by withdrawal of plant growth regulators. That is correct. Somatic embryo initiation in plant tissue culture, it is done by withdrawal of plant growth regulators. Continuation of development and completion of maturation of embryo are induced by the application of abscisic acid and imposition of dehydration period. That is also correct. So both statements 1 and 2 are correct. Then next one. Given below are two statements. One is labeled as assertion and the other is labeled as reason. So assertion A. Maintenance of slightly hypertonic osmolarity is significant during protoplast culture procedure. Correct. The isolated protoplasts are osmotically fragile because they lack cell wall. So maintenance of hypertonic osmolarity is required during protoplast culture because the protoplasts are osmotically fragile because they lack a cell wall. So answer is both A and R are correct, and R is the correct explanation of A. Next one. Population of cells in a developing somatic embryo is. So what kind of population do you get? Or population of cells do you get during development of somatic embryo? So you get a heterogeneous and a synchronous population. Answer is option 1. Next one. Choose the correct statements. Kinetin offsets the inhibitory action of auxin-induced shoot bud formation. That is correct. Auxin inhibits spontaneous shoot bud formation. That is also correct. Kinetin promotes the inhibitory action of auxin-induced shoot bud formation. That is wrong. Kinetin offsets the inhibitory action of auxin-induced shoot bud formation. It is not promotes the inhibitory action. Auxin promotes the spontaneous shoot bud formation. Auxin actually inhibits shoot bud formation. Auxin-cytokinin interaction does not have direct role in root shoot. Auxin-cytokinin interaction has got a role in root and shoot formation. So the correct answer is option A and B. So 3. A and B are correct. Then which of the following is true regarding direct somatic embryogenesis? So direct somatic embryogenesis is without an intervening callus phase. In direct somatic embryogenesis, the callus is first produced. That itself is not correct. Callus is not produced. In direct somatic embryogenesis, the embryo is formed from a cell or group of cells without production of callus. That is correct. In direct somatic embryogenesis, the embryo is formed followed by formation of callus. No, there is no callus formation. None of the above statement is true regarding the direct somatic embryogenesis. So the answer here is option 2. Sorry. Next one. Regarding the role of the following elements in plant nutrition and their physiological function. So these are list 1 has got the elements in plant nutrition and physiological function is list 2. So calcium is involved in cell wall synthesis. Calcium pectate you need there. So calcium is involved in cell wall synthesis. A2. Sulfur is for plant part of amino acid side chain. Sulfur amino acids are methionine. So they form part of the amino acid side chain. So and B is 4. Manganese is enzyme cofactor and potassium is osmotic potential regulation. So A2, B4, C1, D3. Answer is option 3. Given below are two statements. Plant transformation requires the regeneration of whole plant from isolated plant cells or tissues. That is correct. Plant transformation, that is after agrobacterium mediated transformation of plant cells, you need to regenerate the whole plant from isolated plant cells or tissues. Such plant regeneration is conducted in vivo so that the environment and growth conditions can be manipulated for better regeneration frequencies. That is not correct because plant regeneration has to be conducted in vitro where environment and growth conditions can be manipulated for better regeneration frequency. So statement 2 is incorrect but statement 1 is correct. So answer is option 3. One is labeled as assertion and the other is labeled as reason. So assertion. Protoplast fusion is induced by furogen and yield cell types unfused protoplast, heterokaryons and homokaryons. Correct. Heterokaryons are small portions of fused protoplast and are the cells of interest. Yes. So both A and R are correct and R is the correct explanation of A. Then next one. Organogenesis is induced in a callus by. So organogenesis is induced in a callus by auxin-cytokinin ratio management. Answer is option 3. High auxin ratio will result in auxin to cytokinin ratio will result in root formation, and if cytokinin is higher than auxin, then it results in shoot formation. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. During in vitro haploid production, the anthers or the microspores are treated with a higher temperature of 35 to 40 degrees Celsius for a short period before moving the cultures to generally used temperature of 24 plus or minus 2 degrees Celsius. Yes. Higher temperatures induce a stress which triggers the microspores that is destined to form embryos towards embryogenic pathway. Now if you want for in vitro haploid production, the anther or the microspores, they are treated with higher temperature. Now why they are treated with higher temperature is because higher temperatures induces stress, and this triggers the microspores to form embryos. So from embryos you can get in vitro haploid plants. So the answer is both A and R are correct, and R is the correct explanation of A. Then next one. A cross between Hordeum vulgare and Hordeum bulbosum resulted in barley plants with gametic set of Hordeum vulgare chromosomes. Okay, that is correct. By embryo rescue, it should be possible to raise haploids from similar type of crosses in plants. That is also correct. So both statement 1 and 2 are correct. Then next one. The liquid plant tissue culture media that contains labile organic ingredients are sterilized by filter sterilization. So in liquid plant tissue culture, those which are not heat stable components, they are sterilized by filter sterilization. So the most commonly used filter is cellulose acetate membrane filters, and they have asked for the pore size of this particular filter paper. So the filter paper is having a pore size of 0.22 microns. Answer is option 3. Next one. The salts used as macronutrient in MS medium supply. MS medium is Murashi and Skoog medium which is used in plant tissue culture experiments. So the macronutrients in MS medium they supply option C, that is nitrogen, phosphorus, potassium, calcium and magnesium. Answer is C. So the correct answer from the option is option C, that is C only. Then next one. Somatic cell hybridization helps in overcoming reproductive isolation barrier to enable gene transfer between crop plants and its wild relative. Okay. To facilitate selection of hybrids, the crop plant protoplast is stained with rhodamine isothiocyanate and the wild relative protoplast is stained with fluorescein isothiocyanate. Both RITC and FITC are vital dyes that carter red color and green color respectively. What would be the color of the somatic hybrid which can be selected by cell sorter? So, somatic hybridization, it is a process of fusion of somatic protoplast from two different plant species or two different plants. So that is somatic cell hybridization, and it helps in overcoming reproductive isolation barrier, and it allows gene transfer between crop plant and its wild relative. So what they did was they want to conduct a somatic cell hybridization experiment between a crop plant and its wild relative. So they have to select for the heterokaryon after this somatic cell hybridization. So first what they did was to select hybrids, they stained The crop plant protoplast with rhodamine isothiocyanate or RITC and its wild relative protoplast with FITC. Now RITC it imparts a red color and FITC it imparts a green color to the protoplast. Now after somatic cell hybridization the protoplast will fuse and you will get a color which is a mixture of red and green. So that will be what color? It will be yellow color because red and green color on mixing will result in yellow color. So what would be the color of somatic hybrid which can be selected by cell sorter will be yellow color. Answer is option. Then next one. The development stage of microspore more suited for microspore embryogenesis is. So late uninucleate and early binucleate stage. Next one. Binding of plant hormones to membrane receptors of plant tissues cultured in vitro induces a cascade of events leading to biological response by the cultured tissue. This phenomenon is known as, so this phenomenon is nothing but signal transduction. It is not transcription, it is not transgenesis and it is not replication, it is signal transduction. Next one. One of the following is the commercially released somaclonal variant of Indian mustard that was released by Indian Council of Agricultural Research India. So it is Bio 902 Pusa Jai Kisan. Answer is option 4. Match list 1 with list 2. So what did Haberland discover? That you have to find out from list 2. So Haberland, he is father of plant tissue culture. A4 and that is in option 3. So option 3 is your answer. B1 Stewart FC and Reinhardt J. That is first time somatic embryogenesis demonstrated. Edward C. Cocking, what did he do? Protoplast isolation in large scale. Carlson P.S. Smith H.H. and Deering R.D. What did they do? They did somatic cell hybridization in plants. So the answer here is option 3. Next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. For somatic cell hybridization, electrofusion method is recognized as best method as compared to polyethylene glycol or PEG mediated method. Fusion frequency by electrofusion is very high while PEG mediated method of fusion results in heterogeneous mixture of parental type protoplast and heterokaryons. That is correct. So for both, so what they have told is for somatic cell hybridization, electrofusion method is best compared to polyethylene mediated fusion. Why? Because fusion frequency by electrofusion is very high, whereas in PEG mediated method you get heterokaryons and heterogeneous mixture of parental type protoplast. So both statements A and R are true and R is the correct explanation of A. Then next one. The sustainable agriculture could use the properties of legumes to form nodules where the following events take place. Goal of nitrogen fixation is to convert atmospheric nitrogen to ammonia. Okay. Nitrification is a part of nitrogen fixation. Rhizobium fix atmospheric nitrogen. Bacteroids are associated with nitrogen fixation. Choose the correct answer from the options given below. So A is correct. Goal of nitrogen fixation is to convert atmospheric nitrogen to ammonia. Nitrification is a part of nitrogen fixation. Here they have asked regarding the nodules. So we have to see for in the nitrogen fixation in the nodules. So rhizobium fix atmospheric nitrogen. That is correct. Bacteroids are associated with nitrogen fixation. D is also correct. So A, C, D is the answer. Answer is option 4. Then next one. Bacteria associated with phosphate solubilization belong to one or more genera. Bacillus, Rhizobium, Enterobacter. All of the above. Answer is all of the above. Protoplast can be isolated from root, leaves, fruits. All of the above can be used for isolation of protoplast. The statement given below are true for nodules. Cyanobacteria are gram negative. That is true. Cyanobacteria are gram positive. That is not correct. Prokaryotes immobilize the available nutrients. That's correct. Crenarchaeotes dominate nitrogen fixation. That is also correct. A, C, D only. Answer is option 4. Given below are two statements. Statement 1. Nitrogen fixation by rhizobium requires anaerobic environment provided in the living cells of the root cortex. The larger organism is called the host. Mutualism is an ecological interaction between the two species in which both benefit. Okay, statement 1 is correct. Now what about statement 2? The commensalism is an ecological interaction where one species is benefited. That is also correct. So both the statement 1 and 2 are true. The statement given below are true for nodules. Lignified external layers of nodules limit the gaseous exchange. That is correct. Leg hemoglobin binds reversibly to oxygen. Yes. That is why you provide anaerobic environment for nodule fixation. Leg hemoglobin is a protein associated with transition elements. Yes. Leg hemoglobin provides aerobic environment. No, leg hemoglobin provides anaerobic environment. Leg hemoglobin is a protein. Yes. So A, B, C and E are correct. So answer is option 3. So next question. The nitrogenase complex associated with nitrogen fixation shows the following. Constitutes of nitrogenase reductase and dinitrogenase. That is correct. This complex is highly conserved. It is present in the cytoplasm of bacteroids. So all of the above are correct. Now for the next question, that is question number 58. I have a doubt with regarding the question whether they have asked for choosing the most appropriate answer or not the appropriate answer. So which one they have meant by the question is confusing for me. So I am not answering that question. 59. Next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. The ammonia is delivered as a product of nitrogen fixation via a specific transporter to the host cell. Here it is subsequently converted mainly to glutamine and asparagines. The glutamine and asparagine are then transported via the xylem vessel to other parts of the plant. That is correct. Nodule cells contain high activities of phosphoenol pyruvate carboxylase. That is also correct. A and R are correct and R is the correct explanation of A. So answer is option 1. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. Ideal tissue for plant tissue culture is apical meristem. Yes, because you can get virus-free plants. Reason. The viruses are unable to take over control of the host biosynthetic machinery. Auxins in meristem inhibit viruses. So both A and R are correct and R is the correct explanation of A. Then next one is match the following. So induced fusion stimuli. Induced fusion stimuli is electric. Protoplast electric potential is minus 10 millivolt to minus 30 millivolt. Fusogen ionic, that is NaNO3 and high Ca2 positive. Non-ionic fusogen, it is polyethylene glycol. So the answer here is option 4, A4, B1, C3 and D2. Then next one. The nodules of soybean export the fixed nitrogen compounds that contain high nitrogen to carbon ratio. These are urea degradation products. Yes, allantoin or in its acidic form. Both are true. The nodules of soybean export the fixed nitrogen compound that contain high nitrogen to carbon ratio. So these are urea degradation products, yes, and allantoin or in its acidic form. So both are true. So the answer here is option 3. Plant protoplast provide a unique single cell system to underpin several aspects of modern biotechnology. Protoplast, choose the correct answer from the options given below. Protoplast are the cells whose cell walls are removed and cytoplasmic membrane is the outermost layer in such cells. That is correct. Protoplast fusion is a physical phenomenon. During fusion, two or more protoplasts come in contact and adhere with one another. That is also correct. Protoplast fusion may be used to produce interspecific or intergeneric hybrids. Yes. The basic step involved in the production of somatic hybrids by protoplast isolation, fusion and regeneration. Yes. They are the basic steps involved in somatic hybrid production. So answer is A, B, C, D only. Answer is option 1. Next one. Trichoderma are mycofungicides and are. So trichoderma are aggressive agents against phytopathogenic fungi. Yes. Have the ability to survive under unfavorable conditions. Yes. Are efficient in utilizing soil nutrients. Yes. So all of the above is the answer. Next one. Does meristem tip culture require treatment for eradication of microbes? The performance of various treatments is listed. Identify the treatment for getting suitable explant for tissue culture. So, meristem tip culture, it requires treatment for eradication of microbes, and the various treatments they have listed over here. Out of that, which is the best one to get suitable explant for tissue culture? So you can go for both thermotherapy and chemotherapy can be used. So answer is option 3. Next one. The statements given an insight into the effect of rhizobacteria on nodule formation. Choose the correct statement from the options given below. Phytostimulatory rhizobacteria promotes growth by providing nutrients and phytohormones. Yes. Symbiosis assists rhizobacteria in performance. Yes. Genotype of the host does not have any role in the formation of nodule number, density, number and size. Genotype of the host does have a role in the formation of all these things. The genotype determines the phytostimulatory response. That is also correct. So A, B, D are correct and the answer is option 3. Next one. Virus-free plants can be produced through the following tissue culture method except shoot meristem culture. Yes, you can get virus-free plants. Thermotherapy, yes, and meristem culture, yes. Treatment of meristems with thermotherapy, and then you can use the meristem for meristem culture. Yes, cryotherapy, yes. Tissue culture using leaf as explant. No. That doesn't give you virus-free plants. Then next one. Which of the following is the best explant for gynogenic haploid production? So for gynogenic, that is the female reproductive organ. So for these, excised ovules will be the best explant for gynogenic haploid production. Whole floral buds means you will have both anthers and ovules. But excised ovules will give you gynogenic haploid production. Then next one. Veena wants to perform androgenic haploid induction. She wants you to assist her in providing with suitable stage she could select for the selection of explant. Your advice to her is that the best result could be achieved by. So Veena wants to perform androgenic haploid induction, and which is the best suitable stage for that. So the explant just before pollen mitosis will be the best suitable explant for androgenic haploid induction. Answer is option 1. Which of the following is not an inherent problem with micropropagation? Hyperhydration. Yes, that is a problem with micropropagation. Recalcitrance of some plant. Yes. Oxidative browning. Yes, that is also a problem with micropropagation. High rate of multiplication in vitro. That is not any inherent problem. So answer is option 4. Next one. Auxins are associated with elongation, apical dominance and rooting. Some of the experimentally used auxin for callousing includes. So they have asked for auxins involved in callousing. IAA, yes. IBA, yes. NOA, yes. 2,4,5-T and 2,4,5-D. All these are auxins which are involved in callousing. So answer is option 1. Then next one, again the same question. Virus free plants are produced by. So they are produced by shoot tip culture. Answer is option 2. Now this is the third time they have asked a question on virus free plants in the same question paper. Next one. Which of the following is not the basic component of plant tissue culture media? Essential elements in the form of complex mixture of salts. Yes. Organic elements as source of vitamins. Yes. A source of carbon. Yes. Bactericides to control contamination. So plant tissue culture media doesn't use any bactericides to control plant contamination. Next one. Following points are essential for suitable field testing of the micropropagated plant. Supervision and attending to the field should be made all the times. Yes. Collaboration with the growers is essential. Yes. Uniformity in terms of sample or plotted in research station is not essential. That is incorrect. Survival of plants depend on its adaptation before transplanting to soil. That is also correct. Adequate controls should also be used for comparison purposes. That is also correct. So for field testing of micropropagated plant, that is hardening stage they have asked. So A, B, C, D, E, sorry, C is not correct. A, B, D, E are correct. So answer is option 1. Sorry. Next one. Chromosome doubling in microspore culture technique is achieved by treatment of microspores with colchicine. Answer is option 2. Next one. The main use of microspore culture is to provide homozygous lines. That is correct. Then next one. During protoplast fusion, one of the parental protoplast is R or X irradiated and the other treated with iodoacetamide to facilitate only the hybrids to proliferate in the culture medium. The Parents alone will fail to proliferate. Such arrangement facilitates. Okay. So during protoplast fusion, one of the parental protoplast is either X or irradiated and the other is treated with iodoacetamide to facilitate only the hybrid to proliferate in the culture medium. So the parents alone. So out of these two plants, only the parents alone will fail to proliferate, of course, and only you will get only hybrids. So such arrangement. So this type of protoplast fusion, such arrangement, it will facilitate full genome transfer. No. Genome of only one plant remain. No. Partial genome transfer from irradiated protoplast. Yes. From irradiated protoplast, only partial genome transfer will occur. Partial genome transfer from iodoacetamide treated protoplast. Iodoacetamide treatment will not cause partial genome transfer. No gene transfer between the protoplast. That is also not correct. So C only is the answer. Answer is option 3. Then next one. The factors that induce somaclonal variation in tissue culture is or are. Age of tissue culture. Yes. Tissue culture medium. No. Genotype. Yes. Growth regulators. Yes. Explant used. Yes. Whether it is leaf or shoot or root, whatever explant is used, somaclonal variation in tissue culture is one of the factors that induce somaclonal variation in tissue culture. So the answer here is ACDE. Answer is option 4. Next one. Bt based biopesticides depend on the high quality and high efficiency processes. However, it should have one or all of the given features. Palatability of the targets. Yes. Bt based biopesticides. They depend on the high quality and high efficiency process. However, it should have one or all of the given features. Palatability of the targets means it should be palatable. Targets here means the insects. It is a pesticide, Bt based biopesticide. So it should be palatable by the insect. Long shelf life. Yes, that is also correct. Spore crystal complexes which could be easy to use. Yes, that is also correct. So the answer here is option 4, all of the above. Then one of the following statement is mistakenly placed here. Can you help in identification? Phosphate solubilization is not associated with global cycling of insoluble organic phosphate. Organic soil phosphate is formed by various saprophytic bacteria. That is correct. Solubilization of phosphate is associated with pH of the soil. That is also correct. But phosphate solubilization is not associated with global cycling of insoluble organic phosphate is not correct. So answer is option 1. Next one. Trichoderma species have been very successfully used as mycofungicides because they are fast growing with high reproductive capacity. They inhibit a broad spectrum of fungal diseases by diverse control mechanism, are excellent competitors in the rhizosphere, have a capacity to modify the rhizosphere. All of the above. Answer is all of the above. Next one. The biocontrol mechanism in trichoderma may be a combination of different mechanisms which may include the following. Mycoparasitism, yes. Mycophage, yes. Antibiosis, all of the above. Answer is option 4, that is all of the above. Next one. Which of the following are major application of haploids and double haploids? Detection of gametoclonal variation, yes. Detection of recessive mutations, yes. Large scale production of somatic hybrids. No. Fixation of F1 recombinants. Yes. Sorry. Yes, that is correct. Fixation of F1 recombinants is correct. A system to study embryogenesis process. That is also correct. So A, B, D, E are the answer. Answer is option 4. Then TACK super group of plant growth promoting bacteria include the following members. Choose the correct answer from the options given below. A. Garochiota, Cremarchiota, Lochiarchiotas and Taumararchiota. So out of Lochiarchiotas, all the other three are involved in TACK super group of plant growth promoting bacteria. So answer is A, B, D only. Protoplasts are very sensitive to osmolarity of the medium because they lack cell wall. To keep the protoplast intact and facilitate their growth and cell division, the isolated protoplast is plated in a culture medium that has high osmoticum than the protoplast. Yes. The most appropriate high osmoticum obtained is by supplementing the liquid culture medium with. So you have to supply some external source which will provide high osmoticum, and that can be obtained by 500 to 600 millimoles per liter mannitol or sorbitol. Answer is option 1. The next one. Given below are two statements. Statement 1. Ammonia nitrogen is fixed into organic compounds relatively easily because it is more reduced than other forms of nitrogen. Yes. Many bacteria employ glutamate dehydrogenase for ammonia assimilation at high ammonia concentration. That is also correct. So both statements 1 and 2 are correct. Next one. An alloplasmic CMS line. CMS is cytoplasmic male sterility line. Suffers from chlorosis due to incompatibility between chloroplast or alien species and nuclear genome of cultivated species. A method by which cytoplasmic organelles can be manipulated to overcome the problem is. So a alloplasmic cytoplasmic male sterile line suffers from chlorosis because it is having incompatibility between the chloroplast of alien species and nuclear genome of cultivated species. So a method by which cytoplasmic organelles can be manipulated to overcome this problem. So this problem can be overcome by somatic cell hybridization. Answer is option 3. Next one. The most commonly used biocontrol agent are living organism. Yes. Pathogenic for the pest of interest. Yes. Beneficial to agriculture. Yes. Useful for public health programs. That is also correct. So A, B, C, D is correct. Answer is option 3. Next one. Tissue culture or protoplast requires a high skill. Assume you have been given necessary facility for protoplast culture. Arrange the following steps in the sequence to achieve the goal of culturing the protoplast. Choose the correct answer from the options given below. So following step in arrange, you have to arrange these steps in sequence. So first step will be sterile leaf tissue. 1. Isolate and purify the protoplast. 2. Then incubate in cellulose and pectinase enzyme. You have to remove the cell wall. So for that you have to incubate in cellulose and pectinase enzyme mixture for 2 to 3 hours. Then next will be, I made a mistake. One will be sterile leaf tissue. Then you have to chop the tissue into fine pieces in neutral buffer or sterile water. Then you have to incubate in cellulose and pectinase enzyme mixture for 2 to 3 hours. Then you have to isolate and purify your protoplast. Only after treatment with cellulose and pectinase to remove the cell wall, you will get the protoplast. So isolate and purify the protoplast, count the protoplast and plate. So the answer here is B, D, E, A, C, A. B, D, E, C, A. So the answer is option 2. Next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion. Reduction of molecular nitrogen is highly exergonic reaction. Yes. During nitrogen fixation. Okay. Reason. Reaction has high energy of activation due to N triple bond N. Hence nitrogen reduction require large number of ATP molecules. So nitrogen fixation, it requires large number of ATP molecule because of this reason. Because N triple bond N is present and the reaction is highly exergonic one. So both A and R are correct and R is the correct explanation of A. Then next one. Auxin promotes cell division and root differentiation while cytokinin promote growth through regulation of cell division and shoot differentiation. Auxin does not antagonize cytokinin action. Auxin antagonize cytokinin action when low auxin concentration limits the action of cytokinin. So statement 2 is not correct whereas statement 1 is correct. So answer is option 3. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion. 15 to 45% PEG is used for achieving protoplast fusion. Correct. PEG is insoluble in water. It is soluble in water and therefore cannot form hydrogen bond to facilitate bringing together of adjacent protoplast, which in turn spontaneously fuse to form the hybrid. That statement is not correct. PEG is soluble in water. So the answer here is A is correct but R is not correct. Then next one. The zygotic given below are two statements. The zygotic embryos and somatic embryos both typically pass through globular, scutellar and coleoptilar stages. Yes. Somatic embryos show secondary embryogenesis and pluricotyledony, which is absent in zygotic embryos. Yes, that is also correct. So both statements 1 and 2 are correct. Next one. The nitrogen fixation can proceed only at very low oxygen concentration by action of dinitrogenase because the nitrogen used for nitrogen fixation is taken from the air. Yes. The cytochrome play an important role in reducing to minimal amounts. Yes. So both 1 and 2 are correct. So next question. Match list 1 with list 2. Trichoderma harzianum. Trichoderma harzianum is commercial preparation F stop A3. Trichoderma polysporum. It is for control of wood decay. Agrobacterium radiobacter Galex used for crown gall and Pseudomonas fluorescens commercial preparation Drager G. So the answer here is option 1 A3 B2 C1 D4. Then next question. Which of the following is not a molecular marker gene for somatic embryogenesis? It is RGA1, whereas others are molecular marker genes for somatic embryogenesis. The RGA1 is not. Then next one. Gynogenic haploids rather than androgenic haploids are preferred in certain plants for genetic studies and crop improvement. It is certainly preferred in members of Asteraceae, Chenopodiaceae and Liliaceae which exhibit a poor androgenic response, and in cases where androgenesis through albino plants. Choose the most appropriate answer from the option given below. Both statements 1 and 2 are correct. Then next one. Find the statements which are not true for root nodules. Nodule formation is controlled by chemical signals between plants and microbes. Chemical signal which is not true for root nodules. Okay, that is correct. Nodule formation is controlled by chemical signals between plants and microbes. That is correct. Root nodule is a selective parasite between legumes and leguminous bacteria. Yes. Leguminous bacteria receive signal molecules like flavonoids and their glycosides from the host plant. No. Which are not true for root nodules. Legumes also secrete antagonists that promote the flavonoid-mediated activation of nodulation genes. Yes, that is correct. Node ABC gene is essential for nodulation and is common to all modulating bacterial species and strains. So that is not correct. So C and E only. Answer is option 3. Next one. Soma clonal variation results from changes in. So it is a chromosome number. Yes. Chromosome structure. Yes. DNA methylation. Yes. Gene mutation. Yes. And gene amplification. So A, B, C, D, E. Option 4 is the answer. What influences the development of plant in tissue culture? Wavelength, flux density and photoperiod. Yes. Wavelength and density of light. No. Exposure to light, short photoperiod. No. It is wavelength, flux density and photoperiod of light. So that's it. Thank you for watching my channel. If you are interested in writing the life science exam for MSc Entrance of CUCET, you can refer to my channel playlist. I have uploaded life science question papers of 2021 and 2022 also. So that's it. Thank you for watching.